Okay, there are other bio-macromolecules are present. Okay, when you observe here, like proteins, nucleic acids, polysaccharides, and all that. Okay, we'll discuss one by one. Okay. Okay, when you observe here, so lipids are strictly considered as a bio-macromolecules. So when you observe the acid soluble pool, the acid soluble pool represents roughly the cytoplasmic composition. So when you observe the acid soluble pool or fraction, or that is the filtrate that is obtained during the chemical analysis, okay, that is considered as the acid soluble pool. Okay, that gives the composition of the cytoplasm of the cell. Okay, next, the macromolecules from cytoplasm and organelles become the acid insoluble fraction. Okay, whereas the macromolecules, which will comes under macromolecules means, okay, these are also present in the cytoplasm and organelles, that is cell organelles. Okay, but they are present in the acid insoluble fraction. That means the substance which is not filtered through the okay, the filter that we used to separate the molecules. Okay, so together they represent the entire chemical composition of living tissues or organisms. So this acid soluble fraction and acid insoluble fraction all together, they form the composition of living tissues or organisms. Okay, that is it. And next, in summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organisms. So, if you arrange the the chemical composition, if you arrange the if you represent the chemical composition of living tissue, okay, from abundance to lower, okay, from higher abundance to lower abundance, okay, in the cell, okay, which chemical is present in large quantity means water, because water is also chemical, H2O. Okay, that is so water, the most abundant chemical is water. Okay, next to that, other chemical substances are present. That table here it is given. Okay, water is the most abundant chemical in the cell or in a tissues or an organism. Okay, it ranges from 70 to 90 depending upon the cell to cell. They slightly variation will be there. Okay, and that ranges from 70 to 90. And next, coming to the proteins. Okay, the percentage of proteins ranges from 10 to 15. Okay, that is a protein. Next to water, which are present in abundant means proteins. And next, after proteins, we'll find nucleic acid, DNA and RNA. Okay, that is that accounts about 5 to 7%. Okay, because the nucleic acids are present in almost all the living cells, those which are having nucleus. In all those cells, we'll find this nucleic acids, both DNA and RNA together, we call it as nucleic acids. So it ranges from 5 to 7%. And next to nucleic acids, we'll find carbohydrates. The percentage of carbohydrates is around 3%. And next, lipids. Next to carbohydrates, we'll find the lipids, and the percentage of the lipids ranges from up to, it will be up to 2%. And the remaining all other elements which are present in ionic form like sodium, potassium, calcium, iron, zinc, whatever it is, all they comes and below 1%. It is in human, it is usually around 0.9%. In the human cells, it will be around 0.9%. That is less than 1%. So it will say nearly 1%. Okay, that is the chemical composition of different biomolecules that are present in the living cell. The most abundant is the water, which accounts about 70 to 90%. And next, proteins, okay, which ranges from 10 to 15. And next, carbohydrates, which ranges up to 3. And lipids, which will up to 2%. And the nucleic acids ranges from 5 to 7%. And ions is around 1%. Okay, that is the chemical composition of different biomolecules that are present in the living organisms. Okay, and one. So that is water. Water is aqueous, which it will provide aqueous nature to the cell. Okay, all the biochemical activities that are taking place in the living organisms will take place in the presence of water. Water is acting as a medium for all these biological activities. So there is no surprise, okay, in having water as a higher percentage, that is 70 to 90%. So there is, and next one is proteins. Okay, next to water, which is more abundant, means that is proteins. It is around 10 to 15%. Okay, and next, the proteins are also called polypeptides. Okay, proteins are polypeptides. Why we are calling it as a polypeptides means, when you observe what are the structural units of proteins, or what are the structures with which the proteins are made up of, means amino acids. Amino acids are the building blocks of proteins. So when you observe this amino acids, the adjacent amino acids are joined by or binded by a bond called as a peptide bond. As a protein molecule is made up of many amino acids which are bind by many peptide bonds. That's why we are calling it as a peptide bond. Okay, the number of peptide bonds in a protein molecule is equal to the number of amino acids with which that protein is made up of -1. That one will give the total number of peptide bonds that are present in a protein molecule. Okay, how many with which that protein is made up of? -1 will give the number of peptide bonds that are present in that organism. Okay, in that protein. That's why, okay, as they possess many peptide bonds, that's why we are calling it as a polypeptides. Okay, and next, when you observe here, basically the protein molecule, which is made up of amino acids, okay, they are linear chains of amino acids. They are linear chains of amino acids linked by peptide bonds, as shown in a figure. Yes, you can see here. This is one amino acid, this is another amino acid. Like this, a large number of amino acids are joined together. Okay, and we can see one amino acid is joined with another amino acid with the carbon and the nitrogen. Okay, the carboxylic group of one amino acid and the amino group of the adjacent amino acid are joined with the help of a bond called peptide bond. The bond present between carbon and nitrogen, we are calling it as a peptide bond. With the help of that peptide bond, okay, the amino acids are joined, and usually they are linear structures. Okay, primarily they are linear structures of proteins. That's what we can see here. Okay, that is what he is saying. Each protein is a polymer of amino acids. So each protein is made up of many amino acids. As there are 20 types of amino acids. Okay, already we have said very clearly. Okay, in the living organisms. Okay, we'll find a maximum of 20 different types of amino acids like alanine, cysteine, proline, tryptophan, lysine, etc. Okay, and next, a protein is a heteropolymer and not a homopolymer. What is a polymer means? A polymer is a molecule which is made up of a large number of smaller units. A large number of smaller units are joined together to form a long chain called polymer. So when you observe here, molecule consists of two or more different types of amino acids. Okay, two or more than one different type of amino acids. We'll find alanine, cysteine, proline, tryptophan, cysteine, methionine, glutamic acid, glutamine, histamine. Like this, different types of amino acids are arranged in a linear manner to form a protein molecule. So as it consists of two or more number of amino acids, usually the protein molecule is described as a heteropolymer and not a homopolymer. And not a homopolymer. That means it is not made up of only one type of amino acid. So that's why we are calling it as heteropolymer, the protein molecule. So a homopolymer has only one type of monomer repeating n number of times. So what is a type of homopolymer means? A homopolymer is a polymer or polymer which is made up of only one type of monomer. Okay, only one type of smaller units. Okay, there is n number of times. That's why we are calling it as a homopolymer. If only one type. If two or more different types of monomers are joined to form a polymer means, then we call it as a heteropolymer. So definitely protein is a heteropolymer. This information about the amino acid content is important as later in your nutrition lessons, you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. So when you observe, depending upon the source of these amino acids, okay, the amino acids are divided into two groups, essential amino acids and non-essential amino acids. What are essential amino acids means? The amino acids which are not synthesized in our body. The amino acids which cannot be synthesized in our body. And these amino acids has to come only along with the diet we take. Because as they cannot be synthesized, they have to come along with the diet or food we take. Okay, such amino acids we are calling it as essential amino acids. That's why, okay, we have to prefer for the proteins which are rich in essential amino acids. That is the quality food. That one only we say it as quality food. Okay, and the second type, that is non-essential amino acids. The amino acids which can be synthesized in our body even if they are not present in the diet we take. Because they can be synthesized in our body. That is what we can see here. Okay, you will learn that certain amino acids are essential for our health and they have to be supplied through our diet, only through the food we take. Okay, through that only these amino acids are obtained. So hence dietary proteins are the source of essential amino acids. Dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make while we get essential amino acids through our diet or food we take. So those amino acids which can be synthesized in our body, even if they are not present in the diet we take, such amino acids we call it as non-essential amino acids. Okay, the latter are those which our body can make while we get essential amino acids through our diet or food. So protein carry out many functions in living organisms. And next, when you come to the functions of the proteins, okay, the proteins are having a diversity of functions. That means large number of different functions are carried out by the proteins. Okay, some are some transport nutrients across cell membrane. Already, okay, while we are discussing about plasma membrane, we have come across. Okay, in the plasma membrane, we'll find certain proteins. That is integral proteins or peripheral protein. Okay, these integral proteins which are present in the cell membrane, okay, they are involved in the transport of substances from one side of the membrane to the other side of the membrane. So they are involved in transport of nutrients across cell membrane. Okay, those are integral proteins we say. And some fight infections. Okay, infectious organisms are some. Okay, some proteins fight against infectious organism. Okay, like antibodies. Antibodies are also made up of proteins only. Okay, they will help in fighting against the infectious organisms, thus providing immunity to our body. Providing immunity to our body, that is the main role of these antibodies. And next, some are hormones. There are some hormones which are made up of proteins, peptic hormones. And some are enzymes. Okay, almost all or most of the very few, very few are made up of other components. But most of the 99.9% of the enzymes that are present in the living organisms made up of proteins. So some are involved in the formation of proteins. And next, coming to this, the most abundant protein in the animal world. Animal world, remember this one, ma? Collagen. Collagen is the most abundant protein in animal world. Already, while we are discussing about structural organization in animals, we come across the tissues. Okay, in the tissues, we'll find okay, the connective tissues. In the connective tissues, we'll find the fibers, white collagen fibers. And this white collagen fibers are made up of collagen, a type of protein. Okay, and next, the most abundant protein in the total living world, especially related to plants. Okay, the most abundant protein that is present in the plants and in the living world is the ribulose bisphosphate or ribulose 1,5 bisphosphate carboxylase oxygenase. That means this enzyme can act as a carboxylase, that is removes the carbon, and also oxygenase, that means addition of oxygen. So the most abundant protein in the living organisms, all living organisms, that's why it's a whole of the biosphere, all the living organisms, is the ribulose 1,5 bisphosphate carboxylase and oxygenase, which is present in plants, which is very much required for a photosynthesis, or we can say, okay, involved in a carbon This is ribulose 1,5-bisphosphate. Okay, understood? Anybody is having any doubt? Okay. And next, polysaccharides. Okay, where these proteins are present? Proteins are present in the acid insoluble fraction because they are bio-macromolecules and they are having molecular weight usually more than 10,000 Dalton. That's why they were placed under the bio-macromolecules. And the next important bio-macromolecule is polysaccharides. The name itself is indicating poly means many, sacchara means sugar. That's why the name given as a poly, okay? Polysaccharide. The acid insoluble pellet, pellet means solid because it is obtained as solid substance. So the acid insoluble pellet also has polysaccharides, a type of carbohydrate, as another class of macromolecule. As a class, okay, that means, okay, these okay, this polysaccharides, which are a type of carbohydrates, are belonging to the class of macromolecule. Okay, polysaccharides are long chains of sugars. Okay, like protein molecules, which are long chains of. In the same manner, okay, the polysaccharides are also long chains of sugars. Long chains of sugars. Okay, and next there are threads, literally a cotton thread, like containing different monosaccharides as the building blocks. Okay, that is when you observe here, we'll find different types of carbohydrates: monosaccharide, disaccharide, trisaccharide. Okay, and next tetrasaccharide, pentasaccharide, hexasaccharide. Okay, hexasaccharide, heptasaccharide, octasaccharide, nexasaccharide, and finally, if 10 or more number are present, we call it as oligosaccharides, up to 20. And beyond that, we call it as polysaccharides. What are monosaccharides, disaccharides, trisaccharides means? If only one monomer is present, okay, for example, glucose, only one molecule of glucose is present, then the glucose, fructose, ribose, ribulose, xylulose, heptulose, these are all monosaccharides because they consist, okay, they are made up of only one molecule. Okay. And next, disaccharide. Disaccharide means if a sugar molecule is made up of two monomers, that is two glucose molecules, that is maltose. Maltose is a disaccharide made up of two molecules of glucose. And next, the regularly we are using sugar. Regularly we are using sugar, no, for making sweets or any other. Okay, for drinking milk, coffee, tea, we are adding the white color crystalline structure. We are adding, no, that is the normal. Generally, we say sugar or eating sugar. That eating sugar, the chemical name of that eating sugar is sucrose. Okay, it is made up. It is also a disaccharide which is made up of one molecule of glucose and one molecule of fructose. So glucose and fructose are joined by a glycosidic bond to form the single molecule called sucrose. Okay, and next the third one, milk sugar. Okay, the sugar that is present in the milk, that is commonly called, that is called as lactose. Lactose is made up of one molecule of glucose and one molecule of galactose. That means the glucose and galactose are linked by, are are binded by, glycosidic bond to form a compound of two molecules, that is a disaccharide. In the same manner, if three molecules are present, if three molecules of glucose are joined by glycosidic bond, 1, 2, 3 are joined by glycosidic bond, such ones we are calling it as a trisaccharides. Like that, 4 tetra, 5 penta, 6 hexa, 5 7 hepta, 8 octa. Like that, 8 molecules, 9 molecules, and beyond that, 10 molecules and beyond, we call it as oligosaccharides, and up to 20, and beyond 20 we usually call it as 20 to some, many, they are called as polysaccharides. So here we are discussing about the polysaccharides. There are different types of polysaccharides. For example, if you are eating a plant material, for example, if you are eating potato you are eating, what is present in the potato? What is the substance that is present as storage form? In the potato, what is the storage form? Or in plants, how the food is stored, in which form the food is stored? Yes, ma'am. Starch, yes. Starch is a polysaccharide made up of only one type of molecules, that is the glucose. A large number of glucose, up to 900 to 1000, up to 900 to 1000 glucose molecules, one to one, they are linked by the glycosidic bond to form a complex structure called starch. And next, in case of animals, how the carbohydrates are stored? In which form the carbohydrates are stored in animals? Glycogen. Glycogen is also a polymer made up of glucose, but the number of glucose molecules will be less. It will be around 200 to 300. 200 to 300 glucose molecules are joined to one to one with the help of glycosidic bond. With the help of a glycosidic bond. So in the same manner, if you observe the cellulose is also a polymer or polysaccharide which is made up of glucose. So when you observe the starch, which is made up of glucose, and next glycogen, which is also made up of only one type of monomer, that is glucose. Cellulose is also a polysaccharide which is made up of only one type of monomer, that is glucose. So when you observe here, all these are made up of only one type of monomer, that is glucose. That's why these, whatever the polysaccharides, we have said, that is the that is starch, glycogen, cellulose, all these comes under homopolymers because they are made up of only one type of sugar molecules, that is glucose. There is another example for a homopolymer, that is inulin. Inulin, not insulin, inulin, INULIN, inulin. Inulin is a homopolymer of fructose. Inulin is a homopolymer made up of fructose. That means a large number of fructose molecules are joined by glycosidic bond to form a polysaccharide called inulin. Okay, inulin is also a type of storage substance in the plants. Okay, in the plants in which the inulin is present as storage component, in those plants we don't, okay, whatever the carbohydrate they want to store, they store in the form of inulin. They do not store in the form of starch. So if the inulin is present as the storage component, that plants will have inulin. They do not show starch. And next, another homopolymer is honey, which is also a homopolymer of fructose. A homopolymer of fructose. Okay, that is what we can see here. So containing different monosaccharides as building blocks. So first we have discussed about a homopolymer. For example, cellulose is a polymeric polysaccharide. That means a large number of okay, molecules consisting of only one type of monosaccharide, that is glucose. So cellulose is a homopolymer. Okay, starch is a variant of this, but present as a storehouse of energy in plant tissues. And next, animals have another variant called glycogen. And next we can see here, inulin is a polymer of fructose. Is a polymer of fructose. And next, in a polysaccharide chain, say glycogen, the right end is called reducing end and the left end is called non-reducing end. Okay, when you observe a polysaccharide, okay, at one end the hydroxyl group is free. Okay, CH2OH. At one end the hydroxyl group at the fifth carbon, oh sorry, at the sixth carbon is free. So that is that group, wherever it is present, that end we are calling it as a reducing end. But when it comes to the other end, that means it's a long polymer. Okay, at the extreme left side end we have got one hydroxyl group, and at the extreme end of the on the right side, okay, the OH group is not free because it is involved in a glycosidic bond formation. So that end where the OH group or hydroxyl group is not free, that end we call it as a non-reducing end. So we'll have reducing end and a non-reducing end. So you can see in a polysaccharide chain, the right end is called the reducing end and the left end is called the non-reducing end. Okay, the right end is called a reducing end and the left end we are calling it as a non-reducing end. It has branches as shown in the form of cartoon. Okay, for example, if we take cellulose, cellulose is a polymer, okay, branched chain polymer. Okay, branched chain polymer. That means it is not a linear structure completely. It is a branched chain polymer. Okay, and next, okay, as we already said, how many number of glucose molecules usually present in the starch means it ranges from 900 to 1000. So as it is a long linear structure, what happens? Okay, this starch will coil around itself. Okay, the starch get coiled along with it. Okay, as a result, so as the starch is a large molecule and the molecule is folded within it, and the starch is folded within it. Okay, folds within it and forms a helices. Okay, that is a as it is a, okay, as it is a large complex structure. What happens? It folds along, okay, among itself the chain, As a result, it forms a large number of helices, and these large number of helices can hold more number of iodine molecules when iodine is added to the starch solution. As a result, the starch solution from white color, it transforms into a bluish black color or dark blue color. So it is an identification test for the presence of starch in the given sample. So starch forms helical secondary structures. In fact, starch can hold iodine molecules in the helical portion. Okay, the starch iodine. Okay, the starch iodine. Association will give blue color. Okay, in your lower class, you might have undergone this test. Okay, in 9th class or 6th class. Okay, you might have come across this starch test. How to detect the presence of starch in the given solution? Simply add 1 or 2 drops of iodine. Okay, if there is any change in the color from brown color to blue color, it indicates the presence of starch. If there is no any change in the color, then it retains. It says that there is no starch present in that. Why? Because why iodine? Okay, why iodine giving blue color with a starch solution? Why? Because of the presence of a large number of helices. Okay, due to the presence of large number of helices, okay, it can hold more number of iodine molecules, so it will give bluish, okay, blue color with iodine. Okay, but when it comes to glycogen, glycogen is made up of 200 to 300 glucose molecules. But because of the small chain, about 20 to 300 glucose molecules, okay, it cannot hold the small chain, so it will form less number of helices. So it cannot hold more number of iodine molecules. It will hold only less number of iodine molecules. As a result, it will give red color with iodine, the glycogen. The glycogen will give red color with iodine. Whereas cellulose, cellulose does not contain complex helices and hence cannot hold iodine. So, it will remain colorless. Okay, even the cellulose usually does not in water, or most of the polysaccharides usually they do not get dissolved in water. But when you take starch, generally in our houses we can observe. Okay, for the clothes they apply the starch to maintain, to develop a certain strength and stiffness to the cloth that we are wearing. They usually place starch to that. So why? Okay, you can understand. Okay, you can observe how they will place the starch. Okay, first they bring the starch powder, and next they will boil the water. Once the water reaches the boiling temperature, now they start mixing up this starch powder, because the starch usually does not dissolve in cold water. Usually it will dissolve in a lukewarm water minimum. Luke means very slightly warm in nature minimum, that because they will break, because of that heat, it will break the some of the glycosidic bonds that are present in the iodine, in the starch solution, in the starch or in the starch. As a result, the starch is broken down into small molecules. Okay, that is starch forms helical secondary structures. In fact, starch can hold iodine molecules in the helical portion. That's why the starch and iodine reaction gives a blue color. Okay, and next, cellulose does not contain complex helices and hence hold iodine. Okay, when compared to the starch, the cellulose is not having the helices. That's why it does not give any color with iodine. Okay, and here we can see the plant cell walls are made of cellulose. So where the cellulose is present? Cellulose is present as the structural component of the cell walls in plants. And next, okay, this cellulose which is obtained is used for paper making, that is paper made from plant pulp, and cotton fiber is cellulose. That means the cotton fiber. Okay, did you have observed the cotton? Okay, inside the seed will be there, and we'll find some outgrowths from the cotton seeds. These are very fine thread-like structures. They represent cellulose. Okay, this cellulose only is made as a plant pulp for making of paper. Okay, and some cotton fibers is a cellulosic. That means the cotton fibers are also cellulosic in nature. There are more complex polysaccharides in nature. Okay, there are more complex polysaccharides in nature. Complex means if two or more sugar monomers are joined, two or different types of sugar monomers are joined to form a heteropolysaccharides. Okay, they are the build. Okay, they have a building blocks like amino sugars and chemically modified sugars. Okay, amino sugars and the chemically modified sugars. For example, glucosamine and N-acetyl galactosamine. Okay, glucosamine, N-acetyl okay, galactosamine. Okay, these are some of the examples of chemically modified sugars, which are called heteropolysaccharides. And next, the exoskeletons of arthropods, which are made up of chitin. Okay, this chitin is also, the chitin is a chemical substance with which the exoskeleton of arthropods or insects is made up of, and they are made up of with chitin. For example, they have a complex polysaccharides called chitin. The complex polysaccharides are mostly homopolymers. That is, okay, if you observe the starch, glycogen, nucleic acid, okay, sorry, okay, okay, the starch, glycogen, and next cellulose, inulin. Oh, okay, most of these are homopolymers. Okay, most of these are homopolymers. Okay, and the next one is nucleic acids. And the next one is nucleic acids. The other type of macromolecule that one would find in the acid insoluble fraction for any living tissue is the nucleic acid. Okay. So the other type of macromolecule that one would find in the acid insoluble fraction for any living tissue is the nucleic acid. These are polynucleotides. That means a large number of nucleotides are joined by a peptide bond. Okay, sorry, joined by a phosphoester bond, or we can say phosphodiester bond, and results in the formation of a nucleic acid. Okay, these polynucleotides together with the polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. So, okay, we have already discussed about polysaccharides. We have discussed about proteins or polypeptides. So, this is polysaccharides and polypeptides. Okay, they form the macromolecular structures. A nucleotide has three chemically distinct components. One is a heterocyclic compound, that is the nitrogen base. The second is a monosaccharide, that is a ribose. And the third is a phosphoric acid or aspartate or phosphate. Already we have discussed about these nucleotides yesterday itself only. You can see once we go through that material. Okay, as you notice in figure 9.1, the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. So these nitrogen bases are also called as a heterocyclic compound in nucleic acids. Are the nitrogen bases? So there are different types of nitrogen bases like adenine, guanine, uracil, cytosine, and thymine. Okay, the sugar found in polynucleotides is either ribose, that is a monosaccharide pentose, or at a 2' deoxyribose. That means already yesterday I told you very well. Okay, there are two types of pentose sugars that are observed in nucleic acids. One is the DNA and the second one is RNA. In DNA, what is the type of ribose that is present? Means it is a deoxyribose. Okay, while in RNA, we'll find a ribose. We'll find a ribose. Okay, which will make okay ribonucleic acid. DNA is a deoxyribonucleic acid. And next, okay, that is about different biomacromolecules that are present in the acid insoluble fraction. Okay, that is. And next, okay, tomorrow morning you are having the MCQ test. Okay, already I informed you very before. Okay, the syllabus for the tomorrow's test is the different cell organelles that are associated with the endomembrane system. Okay, that is endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. And there are some cell organelles which are not included in the Okay. Endomembrane system like mitochondria, plastids, okay, peroxisomes, nucleus, and all that, because they are not coordinated. The functions of these cell organelles is not coordinated with one another. They were not placed under the endomembrane system. So, okay, you can study about chromosomes, microbodies, cytoskeleton, cilia and flagella, centrosome and centrioles. Okay, all all are included in the syllabus, starting from a endoplasmic reticulum up to the last of the chapter. Okay. Okay, that is the syllabus for the tomorrow's test. Anybody is having any doubt?
No, sir.
Okay, that is for today. If anybody is having any doubt in the examination topic tomorrow or the completed topic today, you can ask.